I'm Professor Chakraborty, and today's lecture is on neuroglial cells. Neuroglial cells are commonly called glia. They are the supporting, protective, and regulatory cells of the nervous system. Although neurons receive all the glamour, and whenever you're talking about nervous system, we're talking about neurons, but glial cells have an equal importance in the nervous system. The word glial, glial comes from the word glue, emphasizing one of their many roles that they bind to neurons together and provide a supportive framework for the nervous system or nervous tissue. They are the silent workforce that ensure that every neural circuit operates with precision, stability, and efficiency. They are similar to if you consider a university, take for example, Adelphi University. If professors and students are the neurons, there are several other workforce that have to work in order for the entire university to work properly, like the sanitation workers, the electricians, the public safety. These are all neuroglial cells that are present, that are required for a successful university to work. Similarly, glial cells are there. They are the silent workers that help the neurons to insulate, support, and nourish the neurons. So, a little bit of background before we proceed. So, this is the entire nervous system. The nervous system consists of two parts: central nervous system, which consists of the brain and the spinal cord, and the PNS, which is peripheral nervous system, consists of the cranial nerves, the nerves that come out from the brain, and the nerves that come out from the spinal cord, which are known as spinal nerves, which we will study in the later part of this course. So, the nervous system consists of two parts: central nervous system, CNS, and peripheral nervous system, PNS. Central nervous system consists of brain and spinal cord, and peripheral nervous system consists of cranial nerves and spinal nerves. And both of them, like they have neurons, they have neuroglial cells. In the CNS, there are four major neuroglial cells. Number one, we're going to go over the preliminary functions of each of them right now, and then we'll go in details as we proceed. There are four major neuroglial cells: astrocytes, oligodendrocytes, microglia, and ependymal cells. Astrocytes are mainly responsible to maintain the blood-brain barrier, BBB. They provide nutrients to neurons. They regulate extracellular ion and neurotransmitter levels. They are very important for repair after injury. Remember, neurons have a very little capacity to repair once they are injured. So astrocytes play a very important role in neuron injury. Oligodendrocytes. They form the myelin sheet, which we learned when we were learning about neurons. They are important for conduction. And one oligocyte is able to myelinate several axons. Microglial cells They have immense immune function and they basically act like janitors, removing phagocytosis debris, pathogens, and damaged neurons. Ependymal cells, they line the ventricles. What are the ventricles? We learn about ventricles in brain at length. But right now, just remember that in the brain, there are grooves that are filled with cerebrospinal fluid, a fluid that is required by the brain. And these ependymal cells line these ventricles and help in the production and circulation of cerebrospinal fluid. They are part of the choroid plexus as well. In the peripheral nervous system, PNS, there are two cells, neuroglial cells. One is called the Schwann cells, which are similar to that of oligodendrocytes in the CNS. They form the myelin sheet. They themselves wrap around the myelin sheet. Each Schwann cell myelinates one segment of one neuron. Also support unmyelinated fibers by surrounding them, and they are also involved in axon regeneration after injury. Very, very similar function like oligodendrocytes. Satellite cells surround the neuron cell body in the PNS ganglia. They are important to produce support and production. They are there for pain signaling and inflammation. So I'm just going to go over for you to take a look at how the physically or how they look under the microscope. The first one, as you can see, the green shown in here are the astrocytes, and you can see they are all connected with the neurons. Oligodendrocytes, the blue structure that you see in here, these are the myelin sheet, and they form these myelin sheet, the oligodendrocytes. Microglial cells, these are there in order to help in the process of phagocytosis. And ependymal cells, these are the ventricles, they surround the ventricles, and these ependymal cells are involved in the formation of CSF, cerebrospinal fluid. Next are a photograph of the PNS. As you can see, these are the neuron, and these are the Schwann cells that actually form the myelin sheet and themselves surround the myelin sheet or the axons. Okay, there are parts which are known as the nodes of Ranvier, that's exposed in there. There are satellite cells. These are the satellite cells around here. They are present to support the cell body. So we are going to start with the first one, which is the astrocytes. So as we said, that astrocytes are there for protection, and they are mainly responsible for the blood-brain barrier. That's the main function. So apart from the blood-brain barrier, they remove excess neurotransmitters, and they are also responsible for glycogen reserves. We'll learn about it in details one after the other. So first is blood-brain barrier. So the question is, what is blood-brain barrier? Blood-brain barrier is a highly selective protective barrier formed between the blood and the brain. Blood carries several proteins, several neurotransmitters, ions, gases, water. So all of this cannot be transmitted or cannot go into the brain. it has to be selective. Why? Because brain is a sensitive structure. It has millions of neurons, and we do not want the neurons to be excited all the time. Or we do not want the neurons to be inhibited all the time. It has to be regulated. In order for it to be regulated, we have the blood brain barrier, a guard between the blood and the brain so that only a selective number of substances can go in. So if you look into this picture, you will see this is the neuron and this is the blood vessel, right? Everyone understands that. This is the shown in green is the neuron. This is the blood vessel. So if I take a cross section of this, and so the longitudinal cross section of it, this is the blood. These are, the green structure is a neuron, and this is the astrocyte. There are three main components of the blood-brain barrier. Number one are these endothelial cells which are present. Then there's a basal membrane which separates it from the astrocytes, and the astrocytes themselves, the foot of the astrocytes are known as pericytes, and they are present in together, they are called the blood-brain barrier. So these endothelial cells, as you can see them, in between these cells, there are tight junctions. These tight junctions are responsible for communication between the blood and the extracellular fluid, between the brain. Blood and the brain, and this is the barrier. I want you to understand the selective permeability of the blood-brain barrier is very important, and it is maintained by the astrocytes. The astrocytes are able to regulate the tight junctions. How do they do the regulation? They regulate the tight junctions. So if they increase the tight junctions, number of tight junctions, they are able to make the membrane more or less permeable so that only the desired substance can come in. And if the requirement is such that we need more of those substances, they can increase the permeability by having more tight junctions so they can move into the brain. The blood-brain barrier is an extremely important structure, and its main function is to protect the brain from toxins, pathogens, and fluctuations in blood composition. It maintains homeostasis necessary for neuronal function. It regulates nutrients like glucose, amino acids, and also certain gases like O2 and CO2. It prevents entry of many drugs and large molecules. So, reviewing this again, blood-brain barrier is a highly selective protective barrier between blood and the brain. It is composed of endothelial cell, basal lamina, and the foot of astrocytes. Their astrocytes are able to produce growth factors which can regulate tight junctions, and as such, it can regulate the permeability of the blood entering into the brain. It is selective in the substances that it can let come into the brain or from the brain to go into the blood. So, one key important factor to understand here is in the brain, there are certain regions where the blood-brain barrier is broken. And these are area of prostema. So if you look in here, you will be able to see that this is the region where around the third ventricle, there is a region which is known as area of prostema, where there are so So here we are. We're talking about a region of the blood-brain barrier, which is the region of prostema. Region of prostema is a crucial part. It is a vascular structure near the brainstem and medulla oblongata, and it is there to detect toxins. If there are toxins, what it will do is it will induce vomiting reflux. As such, you will vomit out any toxins. So that is the reason that the blood-brain barrier is not there. There are several other regions like median eminence, which is in the hypothalamic region, where it controls it because the release of the regulatory hormones from hypothalamus happens. So it has to be, this blood-brain barrier is not there so that everything, all the regulatory enzymes can pass from the hypothalamus to the pituitary. Posterior pituitary is there so that hormones like ADH and oxytocin can be released. It is not there, it's very weak in the pineal gland. And then there are two other regions near the third ventricle, which is known as SFO, which is subformal organ. It is just beneath the fornix and near the third ventricle, and OVLT is also, which is organum vesiculum of lamina terminalis. These are region near the third ventricle where this sense the osmolarity of the the fluid that is coming in so that it can regulate it accordingly. And choroid plexus, because it is responsible for cerebrospinal fluid production, the blood CSF barrier is not there. So this leads us to the next glial cells, which are known as astrocytes. So I know we have not dealt with the synapse right now, but let's understand this little more, in a very fundamental way. So remember, this is a neuronal end or synaptic terminal. So this is the synaptic end and this is the axon of the presynaptic neuron. Pre means because it's sending signal. And this is the dendrite of another neuron, which is known as a postsynaptic neuron. Okay. So signals from here, which are basically vesicles with neurotransmitters, are released into this region, which is known as the synaptic cleft. And these neurotransmitters have receptors which are present in the postsynaptic neuron, which are taken up by the postsynaptic neuron. But there is no balance between the release and the uptake. So the release of the neurotransmitters is usually higher than the uptake. That means the receptors present on the postsynaptic neuron is relatively less than the neurotransmitters that are released in the synaptic cleft. The release of the synaptic cleft, the neurotransmitters, are there in the synaptic cleft, and if they are present continuously, it can lead to several diseases. So there is a mechanism by which the reuptake takes place, and that is helped by the astrocytes. They, for example, if it is a glutamate, it can take up glutamate, convert it in glutamine, and bring it back to the presynaptic neuron to be recycled. Or the glutamate can go into the TCA cycle and be stored as ATP. And as and when needed, it can supply the ATP to the presynaptic neuron or the postsynaptic neuron as needed. So the reuptake of these neurotransmitters are very, very important, and we will learn about several diseases when they are present in there and are not taken up quickly enough, what happens. But right now, just remember, these reuptake of these neurotransmitters are done by the astrocytes, which help to recycle or production of ATP in order for the proper functioning of the electrical impulse to carry on within presynaptic neuron and postsynaptic neuron. This is the second function of the astrocytes. Then the third very important function of astrocyte is glucose uptake. For neurons, glucose is the only source of energy. So glucose is taken up by the neuron, and is converted into pyruvate, as we all know, goes through the TCA cycle, and these glucose can then be converted into glutamate, and then these neurotransmitters are released into the synaptic cleft. So this is a presynaptic neuron, this is a postsynaptic neuron. Now, these glutamate, as we said, if they are present in excess in this region, in the synaptic cleft, can cause several diseases. So they can be taken up by the astrocytes, and then they can be converted into glycogen and can remain stored in the astrocytes. And when needed, this glycogen can again be converted into glucose and can be recycled. In addition to that, glucose can also be taken up by the astrocytes and converted into pyruvate, and pyruvate can be transported to the neurons, and they can use it as a source of energy and can be used to form the entire cycle of events can continue. So it is very, very important that astrocytes help in the glycogen reserve. Again, neurons can only use glucose as their main source of energy. Glucose can be taken up by the neurons directly and can undergo TCA cycle to produce energy, or they can be used to be converted into certain neurotransmitters. And glucose can also be taken up by astrocytes to be converted into pyruvate into lactate, and lactate can be taken up by the neurons. It can also be, it's possible that it can also be converted back into glucose to form glycogen and remain stored in there. Glycogen may be a starting material for formation of certain neurotransmitters. And glutamate, which is a neurotransmitter, if released, they can also be taken up and stored in the astrocytes for future use. Next comes oligodendrocytes. Oligodendrocytes are very, very important because they are responsible for making up they help in the formation of myelin sheet. So if you look in here, you can see these are the myelinated neurons and these are the myelin sheet, and these are the oligodendrocytes. The oligodendrocytes, the counterpart of oligodendrocytes in the PNS is the Schwann cells. Schwann cells also produce myelin sheet, but those are in the PNS. The oligodendrocytes, remember, the myelination of the axons is essential for electrical conduction because it speeds up the process. Only the region where there are the nodes of Ranvier, where several sodium potassium pumps are present, the myelination is not there. Rest, each of them are myelinated. And these oligodendrocytes are able to myelinate several axons at the same time. So, one oligodendrocytes can myelinate several axons. In case of Schwann cell, actually it doesn't happen. Each Schwann cell is able to myelinate only one part of the axon. So that's kind of a difference, which we will do a little later. So here we see that near the nodes of Ranvier, there are no oligodendrocytes, but one oligodendrocyte is able to myelinate several, parts and several axons. So this is the difference between oligodendrocytes and Schwann cell. One common commonality is the function of both oligodendrocyte and Schwann cells is to myelinate the axons. But one oligodendrocyte is able to myelinate several fractions of the myelin sheet in one axon, or is able to myelinate several axons. One oligodendrocyte, several axons. But in case of Schwann cells, each segment is myelinated by one Schwann cell. Remember, these Schwann cells not only myelinated, they actually wrap themselves around it to form which is known as neurolemma. Okay, so these are little different in the way they actually myelinate the axons. This is basically showing you the differences between oligodendrocytes and Schwann cells. There are certain common factors and then there are certain differences. The common factor is both are responsible for myelination of axons. Oligodendrocytes are present in the CNS. Schwann cells are present in PNS. Myelin, one oligodendrocyte is able to myelinate many axons. Schwann cells, one axon segment, one Schwann cell. Regeneration of the CNS with oligodendrocytes is really poor, but with Schwann cells, it's excellent. So that is the reason when a neuron gets damaged in CNS, becomes very hard to regenerate it. But in the nerve system, in the PNS, if any damage happens, there is a high probability that it can be regenerated. Repair environment is inhibitory, and in this it is supportory. In oligodendrocytes in the CNS, there are if there are certain diseases associated with myelin sheath degeneration, which is known as multiple sclerosis, Multiple sclerosis or MS, the oligodendrocytes and the myelin sheet of the CNS deteriorate and are replaced by hard scar tissue. So these scar tissue are unable to transmit impulses, and that can lead to multiple sclerosis, which leads to double vision, blindness, speech defects, neurosis, tumors, and patients really have a hard time knowing the reasons for it. It is very uncertain why this happens, but it does happen. There is another clinical applications, another disease associated, which is known as Tay-Sachs disease. It is a hereditary disorder which usually affects the children, and this is by the production of a glycoprotein known as glycoside in the myelin sheet. And this accumulation of glycosides makes the transmission of impulse very hard and leads to Tay-Sachs disease. Again, there is the patient suffers from blindness, loss of coordination, dementia, and usually happens early on, and victims die by the age of 3 to 4 years. The myelin sheet produced by oligodendrocytes is compact, small nodes. These are thicker, larger nodes. And when the Schwann cell wrap around it, they are known as neurolemma. The fourth cell is an ependymal cell. And if you look in here, this is the brain, and we're taking a cross section of the brain with the blood vessel. Okay, so if you look into this structure, so this part in here is the blood vessel, as you can see, and these are made up of cells, which is these ependymal cells. These are the ependymal cells, and these ependymal cells are fenestrated, means they have gaps in them, and they are part of this BCSFB means blood, CSF means cerebrospinal fluid, B means brain. Blood CSF barrier from the brain. Okay, blood CSF barrier. So from here onwards we are talking about the CSF that rotates around the entire brain, and then there is ventricular barrier, ventricles. We will, as I said, these are grooves, and then this is the brain, and again there is blood. So these ependymal cells actually line the ventricles, and they are responsible for production of CSF. The blood is here. They have fenestrated, ependymal cells have gap junctions. So only certain particles can go from the brain to this region in here, which is the region of the ventricles. And in here, there is production of CSF, which is nothing but a part of the brain. There are certain proteins that are removed, certain other factors like hemoglobin doesn't pass through, certain large molecules doesn't go through, and the formation of CSF is formed, and this CSF has multiple functions in the protection of the brain. So these ependymal cells help in the production of CSF, which is in turn responsible for protection of the brain. So these ependymal cells, which line the ventricles, are very important for the brain function. And the last one are the microglial cells. Microglial cells, as you can see, these are the microglial cells. And the microglial cells have two major functions, actually. Number one, they have an immune function, And number two is they are like janitors. They remove the debris and pathogens from the neuronal vicinity or environment. So as you can see in here, I've shown in the microglial, there are two types of microglial that are produced. One is neurotoxin microglia and the other is neuroprotective microglia. These neuroprotective microglia and neurotoxic microglia produce what is known as cytokines. Cytokines are chemicals which can be of two types. One can be pro-inflammatory and the other is known as anti-inflammatory. Pro-inflammatory are the ones, they help in the formation of inflammation. Anti-inflammation are those which stops the inflammation. So if this is the neuron, as you can say, this is the cell body, these are the dendrites, this is the axon, this is the myelin sheath, these are the axonal terminal. If there is any damage, say for example in the axonal end, what will happen is these they will, in order to reduce inflammation, these neuroprotective microglia are going to help them in the repair mechanism. So once there is a damage, there are debris form, and definitely there is some sort of a pathogen attack or certain virus infection, and those viruses are taken up by the microglial cells. Okay. Apart from that, for neuroprotection, they will produce anti-inflammatory cytokines in order to repair it. These neurotoxic microglia are present to remove the debris, and also further, if the cell completely dies, it will go into phagocytosis. That is, it's going to eat up the cell so that that neuron does not infect any other neurons. So this actually shows two types of functions uh, of the microglial cells. As you can see in here, there are 8 different forms that it can help. The number one is it is involved in neurotransmission. So this actually summarizes the key roles of microglia, the resident immune cells of the CNS. They are constantly performing surveillance, monitoring the brain environment for injury and pathogens. And microglia carry out phagocytosis. If there is a damage, debris, pathogens, they always try to eat them up by the process of phagocytosis. They can actually also modulate neural excitability, influencing whether the neuron should be excited or remain quiet. They have also shown to be performing synaptic pruning, meaning removing weak or unnecessary substances from the synapse. They help in the repair and maintenance, like we just discussed. They also help in the neurotransmission. They influence the neurotransmission by interacting with astrocytes in the synaptic cleft. They are involved in the blood-brain barrier permeability, ensuring proper barrier function under normal or disease states. And they are also involved in inflammation by the release of cytokines, by balancing pro and anti-inflammatory signals. So these are all the functions of the microglial cells. One other function of microglial cells, which I want to discuss a little bit, is in the HIV, human immunovirus. HIV virus infects microglial cell where it can remain silent. This is the silent form, latent infection. It can, infect the microglia and remain silent. Under certain condition, it can get reactivated, leading to activate the HIV virus, which can be renewed viral production inside the microglia, which is this in here. Reactivated microglia release inflammatory cytokines. They release inflammatory cytokines that contribute to neuronal damage. Inflammatory cytokines are, pro-inflammatory cytokines that can cause neuronal damage. So in HIV, that can affect the brain and cause neuronal damage, where microglial cells play a very important role. This actually ends the neuroglial cells. I hope you have understood it and understand the different functions of the neuroglial cells in the CNS and PNS.